food production, and the project is organized in two different parts. The first part is a microbiological part where we select a strain to be used as elite inoculant for alfalfa. And the second part of the project is a part dedicated to new feeding strategy. So is an animal nutritional part. Marcello is the expert of this part of the project. The first work package of the project was dedicated to the dissemination and the communication of the results obtained. So the main actor of our project are three: the alfalfa, the bacteria, the Synorhizobium meliloti, and the ruminant, the sheep. In the first work package, we have selected the strain to be used as inoculant for alfalfa. And we have selected this strain on the basis of two main features. The first is the competition with the other Synorhizobium bacteria present in soils. And to do this, we have analyzed the genomics feature and also the ability to ride toward the roots in order to colonize the nodule. The GWAS analysis permits us to identify that the competition is characterized by a multiple genetic determinant. So it's a very complicated system of gene is involved in this treat. So we have isolates strain from salt soils in Algeria, able to grow in presence of different salt conditions, because the soil salt is one of the main problems in the Mediterranean basins. Then we have tested different cultivar of alfalfa for the ability to grow in presence of increasing concentration of sodium chloride, and subsequently we have tested in combination the two cultivar that we have selected for the characteristic to grow in stress condition. This do the cultivar marina and the cultivar etrusca in combination with the Synorhizobium isolates, able to stimulate the growth of the plants and able to grow in presence of salts. Our results showed that several strains were able to grow to boost the growth of alfalfa, and this was possible also in presence of salts. Moreover, our results indicate that different cultivar was stimulated by different strains. This is a very important result. So we have, on the basis of the results obtained, we have developed the inoculant, testing three different carriers: perlite, compost, and biochar. We test in field trials our strain using cultivar marina, 5 different strains setting on the base of the above mentioned features, and the compost as carrier. There are some pictures from the in-field trials in Italy, Algeria, and Morocco, and these here you can see some results, the main results, where it's possible to note that two strains GR4 and one Algerian strain were able to boost alfalfa productivity. So, concluding this part of our presentation, we can conclude that genome-wide association permits to identify the gene involved in competition. Very important is the study of the combination cultivar bacteria, because different cultivar were boosted by different bacteria, and the strain that we have isolated and tested in field are the potential as efficient commercial inoculant. Please.
Thank you, Carlo, and good afternoon. For the work package 2, the aim was to find the best combination between alfalfa and other dietary ingredients not in competition with food. So, in order to achieve this objective, we selected a number of byproducts, co-products, and leftovers from the Mediterranean area. We characterized these ingredients by in vitro techniques for the nutrient composition and the possibility to combine in a mix efficient in order to meet the nutrition requirement of animals. After selecting the best mix, we tested the mix in an in vivo feeding trial. Okay, by using 24 dairy ewes and divided in experimental control, and we tested a mix of different kinds of co-products and byproducts previously tested in vitro, and we compared these ingredients mixed in a concentrate feed with a traditional concentrate based on ingredients that are in competition with food, such as barley, soybean meal, corn meal. And during the experiment, we evaluated the methane emission, the production and composition, and also the efficiency of this dietary strategy in terms of human edible feed conversion efficiency. The amount of energy and protein provided by the milk produced with the energy and protein in competition with the human. So the amount of energy and the protein included in the diet of sheep, how they convert in energy and protein edible for human. So to do this, we assigned a specific value in terms of human edible fraction for each ingredient. For some ingredients, the human edible fraction is 0, such as in alfalfa A. For other is 20%, and for other, for the ingredients in competition with human is more than 50%, such as in soybean meal, corn meal, and barley meal. So according to this approach, we find that no difference in milk production and composition, and this is very good because the dairy sheep produces the same amount of milk and quality by using a feed not in competition with food. And as regard the human edible conversion efficiency was very efficient because was higher than 1. This means that they produce more protein and energy than the amount of protein and energy included in the diet. And for the traditional concentrate, this is less than 1 or negative, so this means that they are less efficient. As regards the methane emission, it's comparable. This is not significant. The difference between the experiment and experimental and the control diet. And we also assessed the life cycle with a life cycle assessment, the carbon footprint of the diets. Obviously, the experimental concentrate was more sustainable in terms of carbon footprints of diets because a lot of the ingredients were not were byproducts or co-products. So the concluding remarks for work package 2 is the comparison between experimental and control diets resulted in similar meat yields. There was an increasing efficiency in nitrogen use of the dairy sheep, and also an increase of beneficial fatty acid in milk. This means that for some kinds of animals, especially the animals in the second part of lactation, may be very useful to adopt this kind of concentrate because they are less competitive with the human resource. Finally, the last work package is for the dissemination and communication, was based on 4 tasks. The first 2 were about dissemination plan and the communication activities, where we have a lot of activities by using the social media and also the internet website. We organized also a summer school with the collaboration of 2 Italian scientific society, the Italian Scientific Society of Microbiologists and of Animal Scientists. And we produced also as educational activities a motion graphic video translated in Italian, French, Arabic, and in English, in order to communicate also to children the results of the project. Finally, we involved also some stakeholders, in particular Continental Semensis, which is a stakeholder involved in the supply seed of Medicago sativa alfalfa, and they are very interested in the developing market level the result of the projects. So that's all. Thank you very much for the attention.
We do have time for 1 or 2 short questions, not so many questions, but 1 or 2 we can manage, I think. Also, maybe the online people have questions, but tell me, or just speak up. Is there any question? Anybody? We explained everything so far.
Okay.
No questions online.
Thank you.
Okay, so in the interest of time, I would say we move on. If somebody has questions, you are there, you are here, so we can ask you also during the coffee break. Thank you very much. A project to follow, or there is a question one?
Closer to the microphone.
The microphone is over there. Okay, I can go closer to the microphone. Yes, so we will now hear the second presentation of this session, which will be poultry insect. So we keep the same order than before. Just let me go to presentation mode. Yes. So the floor is yours.
Thanks. Hello again. I am presenting you the poultry insect project, which has been closed already successfully. It is about using live insect larvae to improve sustainability and animal welfare of organic chicken production. At the quintessence of the project stood the circular economy and its principles, meaning that we want to take advantage of the huge amount of waste which is produced by agri-food processing in order to use it as an effective substrate for insect rearing and breeding. Insects are a very effective biorefinery hub, delivering proteins, oil, chitin, and also frass, which gains a new value nowadays in the fertilization regimes. And to provide part of these macromolecules to poultry farming sector, but also to aquaculture, and also to generate different bioactive compounds, fatty acids, chitin, and so on. Currently, the most researched and the most used insects for feed for food and feed, I would say, are the BSF, the black soldier fly, the house fly, and the yellow mealworm. And you see there are days to weeks in the life cycle, so it's quite quick, the whole process, the whole growth cycle in breeding regimes. Nonetheless, currently there are 7 insect species allowed in Europe for as feed for aquaculture, poultry, and swine animals. So BSF, the housefly, the yellow mealworm, lesser mealworm, house cricket, banded cricket, and field cricket. But recently, according to the regulation of 2021, also the silkworm can be used, can be processed in animal proteins for aquaculture, poultry, and pig feed. So it is expanded to 8 authorized species. If you look at the EU legislation, very nicely summarized by the EPIF, maybe you know EPIF is the International Platform for Insects as Food and Feed. It's an association of a pan-European association comprised or consisting of mainly industry, but also academics. You can see that the live larvae can be used in most of the animals targeted by the human civilization. Last for cattle, but in cattle you can use protein hydrolyzase. This is poultry insect project in a nutshell. There are agri-food residues a lot. 30–40% of the agricultural and food processing is delivering organic waste. Most of them can be used by BSF and other insects as a breeding substrate, and this can be the biomass of the insect can then be used directly, live larvae by poultry, so by chicken, turkey, and so on, to improve their welfare and their growth efficiency. And this can be validated, and we've done it in poultry insect by analytical analyses, also by sustainability assessment. Yeah, and the insect frass, of course, can be used as a fertilizer for growing plants or crops. There were several experimental diets in poultry insect. I will present you only a few. This was the first design where we have used medium growing breeds. You see the legend at the bottom, what CM, LM, LF, and CF mean. There were 4 experimental groups, 10 birds per pen, 6 replicates, 60 birds per treatment, and in total we had 120 males and 120 females. We have supplemented the the diet with 10% Hermetia illucens on the daily feed intake. This was another experimental design, and here I will go more into detail because this is more interesting. There were 3 experimental groups, also 10 birds per pen, 6 replicates, 60 birds per treatment. The target animal was a local breed in Italy, Bianca di Saluzzo. It's a slow growing breed, with a growth cycle of 150 days. There were 120 males in the group. And we have used control, sustainable diet, and the live larvae separately. Here also the supplementation was a bit more. It was 15% Hermesia illucens. And here you see the details of the diet. So the diet composition in the control, you can see the soybean dominates, and in the sustainable diet we have used mainly protein supplying crops like field bean meal, pea meal, barley meal, but also sunflower press cake or gluten meal from corn. Yeah, here you can see the materials and methods used. Everything legislation conform and ethics approval. Gross performance results have been published in high-ranked journals. One was the black soldier fly larvae used for environmental enrichment purposes, and the question if they can affect the growth, slaughter performance, and blood chemistry of medium-growing chickens. This was published in Animal. Then animal welfare results were published as well. It's submitted basically regarding details of organic medium-growing chicken fed live black soldier fly larvae. It's an improvement study about the welfare improvement. Also, slaughtering meat quality parameters have been assessed. Here you see some pictures. It's everything standardized. Regarding the meat quality parameter results and slaughtering. Also in sensors, there was an open access article which can be read with the title "Unraveling the Chicken Meat Volatilome with Nanostructured Sensors: Impact on Live and Dehydrated Insect Larvae Feeding." And of course, the environmental impact results have shown that there are positive benefits of adopting insect biomass as an alternative to the conventional or even to the improved diets, and this has been also published already. Then we have performed also consumer survey results in several countries: Italy, Norway, Germany, and Belgium. Data from Italy, Norway, Germany, and Belgium reveal significant variations between consumer behaviors and perceptions regarding poultry and the related products. Italian and German consumers showed a higher acceptance of poultry fed with insects, while Norwegian and Belgian consumers were more skeptical. Italian consumers exhibited the greatest food involvement and the lowest food neophobia, while Belgians showed the lowest tendency to introduce new foods. However, chicken and birds are by nature insect consumers, so to say. And this is the team who were involved in the project. You can find the contacts, the website. We have also Facebook and Twitter. And I don't know, Francesco, where did you find this picture about me? But I think it was in the Hungarian Posta somewhere last year. Thank you very much. If you have questions, let me know.
Yes, thank you very much. Any questions? We have a bit of time. We were perfectly in time, I think. So we have a bit of time for questions, yes?
Thank you. Yeah, we have the habit of introducing ourselves in other sections.
Yeah.
From Western Cape University, Cape Town, South Africa. I'm interested to know what you mean with welfare opportunities.
My colleague is online and maybe he can answer. I'm the presenter. He's the expert on nutrition and so on. So welfare. Francesco? Maybe.
Yes, I'm here. Can you hear me?
Yes.
Can you tell us the improvement of the animals' welfare?
I don't understand very well the question because it was a noise. If the question is which type of welfare indicator we have utilized, is this one the question?
Should I say
No, you can tell it again if the question.
Yeah, what do you mean by welfare? How do you test that? You said they showed improved welfare. What does that mean?
Maybe you can switch off the microphone while you speak, otherwise that's
If you show me.
Switch off the microphone.
what we have tested as welfare indicators is we have collected feces of the poultry, and we have tested corticosterone as a biomarker of the welfare corticosterone, and also we have performed some video recording in order to see if the poultry receiving the insect larvae are more quiet than the other one when the operator goes inside the pen. So there is some test, ecological tests that are called avoidance test. And so we have just performed both chemistry and laboratory analysis, but also some ecological tests on in the pen when the poultry are reared in the facility.
Okay, did that answer the question? Yes, good.
Interesting.
Yes. Okay, any other questions? Yes, Liliana, please.
Why were there no females included in the second experiment with the local breed and breed?
Do you want to answer? No. Francesco, can you answer why there were no female laying hen? No, not laying hen. Female hens involved in the Italian breed?
The female. And sorry, but because I cannot understand, I cannot hear the question because in the room is the speaker is far from the microphone. The question is if the female.
Just say it again.
Why were there no female chickens in the second experiment with the local breed in Italy?
If it's possible to come to the microphone to tell directly the question, otherwise I can reply by the chat. Okay, now I read the chat. No, we have not put females in the second experiment because males are usually utilized to produce meat. So we have focused the second trial just on the meat because we wanted to analyze if there is some differences in the quality meat parameters. Because in the second trial we provided the larvae to the poultry for a long time. It's a long time period. So we have just selected the males because the aim was to, apart from the welfare, also was to analyze the meat quality. And the males usually are from the poultry farmer utilize male to produce meat because they have a growth rate higher than the females, so usually are utilized to produce meat. The males are utilized by the poultry farmers. Sorry, I just now read the question.
Thank you.
So I changed now the microphone settings. Maybe you can hear us better now.
Seems he's not
Maybe,
hearing us.
maybe not.
Not at all.
Can you still hear us? Great. Change it back.
Yeah, just fine. It's just when people in the room are speaking, so not you.
Okay, yeah, please.
So you
Yeah.
could just ask.
Okay, so you can hear us. Good.
on the consumer side, you said that the acceptance was lower in Germany and in Belgium.
No, in Norway and
or in
Belgium.
Norway. Did you also ask consumers whether they know what the poultry is fed normally? Because I would doubt that consumers know what the chicken may eat normally.
Yes, we have told them that normally they are eating insects as well in the nature, yeah. But in some cultures they stick to the poultry which is fed with normal animal feed. So they are not so open to that part. We don't know why. Maybe it's because of the insect when they hear the term insect, you know, or we don't know the reason for that. But we have mentioned. So in Germany and Italy they understood. So.
Can I just ask, so if you say the live insects, were they only in the larvae or was it also
Larvae.
Only the larvae?
Larvae. So there was a separate larvae. Yeah, there were different diets with addition of larvae. If you saw, there were 10% addition or 15%. So there were different diets. So not only the live larvae.
Okay,
we have replaced soybean and stuff,
yeah,
which was not desirable, like larvae.
So thank you very much. It's obviously an interesting project. Everybody asks a lot of questions about it. It's also a kind of very hot topic in Germany at the moment, the eating of insects and things like that.
I have some at home.
Yeah, very good.
Some
okay,
samples.
So let's go on to the next project. It's the project SysOrg. Let's me just try to. So, yes, please.
All right, thank you. Good afternoon. I'm Liliana from the University of Kassel and presenting the project Organic Agri-Food Systems as Models for Sustainable Food Systems in Europe and Northern Africa, or shortly SysOrg. The project is also finished by now, and for the projects that are finished, we've been asked to follow this outline, which I will do. So first of all, the story behind the project. Why SysOrg? I've mentioned in my pitch talk that the need for transforming the food system is no longer questioned, but the exact pathways of how to go about it, how to transform the food system is exactly what is being the subject of debate. And this is what SysOrg kind of picked up on and tried to actually contribute to increasing sustainable food production and consumption and facilitating the very shift towards more sustainable and resilient food systems. In order to do that, we actually attempted to identify entry points for a successful design of the pathways for increasing sustainable food production and consumption. And initially, we came up with the assumption that overall there would be common intervention and entry points in food systems for enabling these very transformation processes, which would be generally transferable, but they should be adapted regionally based on the specific context. The questions we asked ourselves at the beginning of the project were, what is the exact understanding of sustainability in order to actually drive the transformation process? What are the reasons, motivations, drivers, and barriers for actors to opt for more sustainable solutions in the food system? And what would be the most promising entry points for developing, consolidating, and disseminating organic food and farming, reducing waste, and shifting to sustainable diets? These 3 points were namely the 3 perspectives out of the 4 that we investigated in our project. So coming to the key findings, on the right, you can see in the top part, you can see our 4 perspectives, which was the system transition, sustainable and healthy diet, reduction of waste, and organic food and farming. And we have analyzed those 4 perspectives in our 5 case territories in Europe and Northern Africa, which was the municipality of Copenhagen in Denmark, the federal state North Hesse in Germany, Warsaw City in Poland, Cilento district in Italy, and the province Kenitra in Morocco. We analyzed, as I mentioned in the pitch talk, or focused on 2 units of analysis, households and the organized initiatives in each of those case territories. And we managed to actually collect over 2,000 survey responses from the households, as well as we interviewed 17 organized initiatives across these 5 case territories. And here are briefly summarized results from the 4 perspectives. I don't know if you can see, within the diet perspective, we did focus, for instance, also at the national level. And here the focus were the food-based dietary guidelines in each of the 5 case territories. And here we were able to observe a large discrepancy, starting from the fact that Morocco, for instance, doesn't have any at all. And the other territories have a very wide range in terms of diversity of their scope, as well as level of detail. For instance, from 1 page to 200 pages. What was, however, common in all of them was the overall advice to reduce meat consumption and to increase plant-based foods in a diet. Looking at our results from the household survey, we were able to see an overall healthier dietary pattern in Copenhagen and Warsaw, whereby looking at our Mediterranean case territories in Morocco and Italy, we were able to see an overall low adherence to the Mediterranean diet, the phenomenon that is widely discussed in the literature. On the positive note, we were able to actually establish that there is a high willingness to change towards a sustainable diet on the side of our surveyed households in all case territories, and the exact aspects that the households are reporting to be willing to change would be the incorporation of more seasonal and local foods into our own diet, as well as meat reduction. Within the organic perspective, our territories were actually on a varying stage of their organic transformation or the overall development of the organic sector, with Copenhagen, Cilento, and North Hesse being more advanced in this regard, and Morocco, Kenitra, and Warsaw in Poland, rather at the beginning stage of the organic transformation, which was well reflected in the survey results, where we were able to see a higher overall declared share of organic foods in a diet in those territories with the advanced transformation of the organic sector, such as North Hesse and Copenhagen, whereby, for instance, in Morocco and in Poland, the share of organic foods in the diet was rather low, the lowest actually. On the positive note, there was a strong association between organic food and sustainable food. However, all these household respondents did actually report price to be #1 barrier towards increasing organic foods in their diet, regardless from the stage of the organic transformation of a specific territory. While we look at the increase in organic consumption over the past 5 years, which was one of our survey questions, here it was observed in all case territories except for Kenitra. Our waste perspective results, we were actually also focusing at national level here as well. Specifically, we wanted to see the existence of certain food waste reduction policies or perhaps programs in the case territories. And here all the territories were quite active in that regard, except for Morocco. And we were able to quantify the self-reported, I have to stress, food waste in the households. And the mean was 399 grams per household and week, or 153 per person and week. However, these values were actually dependent on the family size, meaning that, for instance, Morocco had the highest number of family members within a single family, and therefore the highest per household food waste, but the lowest at the same time per person food waste, which is also an interesting finding. The lowest estimated food waste levels were observed in North Hessia and the highest in Kenitra. However, the typology was different and it seems to be territory dependent. For instance, we had the dominant food waste type being actually partly used foods in Copenhagen as well as Warsaw. And we did have, for instance, dominating waste group being or type being, for instance, leftovers in Kenitra or leftovers after storage in North Hessia. Within the transition perspective, we were able to actually find a sizable number of initiatives active in each of the territory, with the majority of them being active in more than just one single activity. And activities ranged from, for instance, promoting a specific diet to educational and awareness raising campaigns. And we also asked the initiatives to name some barriers and drivers for their for performing their activities. And among barriers, what was interesting, for instance, food culture was named in pretty much all the territories, but also specific infrastructures or also prices for more sustainably produced products. As for drivers here, it is interesting to note that overall raised awareness towards the necessity of sustainable development were, for instance, named, but also civil society or also other initiatives as drivers. And overall, all the initiatives in all the territories emphasize the essential role of collaboration in order to drive the transformation in their territories. So based on the results, we were able to propose certain recommendations for the target groups of our project, one of which were actually policymakers and planners. And here, of course, there were many more that we proposed. But for instance, looking at the food waste policies, we would propose that they start also focusing on household food waste behavioral changes, as well as specific strategies for retailers, food companies, and private households for actually preventing the food waste in the first place, as opposed to, for instance, just focusing on reducing the existing food waste, as it is the case right now. We would also propose facilitating community engagement on territorial level so that the local population becomes more proactive and engaged in the activities on territorial level. And also there is a crucial role of exchange between the local and regional policymakers and planners within the whole policy cycle that needs to be emphasized, meaning all the way from developing policies to their implementation, and especially between Europe and Northern Africa. Looking at some practical findings, we would say that there is a need for integrating various perspectives in awareness raising campaigns and specific activities. For instance, linking educational activities about food waste with other sustainable habits, for instance, dietary ones. Then also bridging local level initiatives with other relevant levels. Because local initiatives were something that was in focus of our project, but the initiatives actually named other levels as crucial for collaboration and for bringing about the change. Also, we would suggest that tailored education campaigns could be developed in schools, which would be important to start from early on. And that also all the awareness raising campaigns better integrate dietary guidelines as compared to how it is done right now. Also, we would suggest motivating general population to become proactive and utilizing university structure for educating not only the students, but also the people outside to the university structure about sustainable diets, strategies for reducing food waste, as well as organic food and farming. So what's next? We have finished the project, and at the end of it, we realized that there is a need to advance further theoretical framing as well as methodologies enabling cross-case, inter and transdisciplinary analysis of territorial food systems in order to enable sustainability transformation. This is a very complex endeavor, and a 3-year project cannot actually fully accomplish the task. There is also a need for further cross-case interdisciplinary investigations in other territorial, organic and agroecological food systems in other contexts in order to be able to actually have more transferable results, and therefore it calls for follow-up projects embarking on this mission. And my big question to you, as we were encouraged to ask, is what does it actually take to get the general public more engaged and proactive vis-a-vis the efforts to help transform the food system? A simple but big question. Thank you for your attention, and here's our consortium and contact information.
Thank you very much. We already have the first question on this. Very nice talk. I'm from the EU in Germany, but last time also by Bonn University, and Ghana we focus a lot on sustainability and consumption and so on. And we had a nice project where we investigated how can we actually educate students in homes, food waste. What we have learned is education is more final solution. And my opinion is, and I hate saying this, you have to put forward people legislation if you don't behave the right way. Pay. I hate saying this. In my opinion, that's the only way to get things paid. What is your opinion?
This is something I generally discuss with my students in one of our modules, which is actually called Sustainable Diets. And yes, this is something that I also kind of am favoring, because looking at certain behavioral trends of consumers and certain negligence, I would say, sometimes it does take this hard measures in order to just trigger some change, like a first impulse, and then it actually does facilitate this mindset change, if you will. So yes, I'm with you on that part.
Yes, of course. What are these hard measures? I'm curious to know more about them.
Taxation, for example, or fines. I think one example is, for example, in Canada, for waste separation, if the households don't properly separate their waste, at that point it doesn't get picked up, and next time they have a fine, the bill to pay. So it does actually lead to behavioral change vis-a-vis waste separation, which is just one example.
Sorry, could you speak up because they can't hear the question in the room? So if you could speak up.
So more or less the carrots and stick approach, where you provide incentives, you raise the costs of certain behaviors or bring certain behaviors. So if your costs are inclined to continue to do those things, or you give them subsidies to encourage certain environmental friendly behavior. So basically raising the price of everything.
Let me rephrase my question. Would you have any suggestions beyond the 3 issues of taxes, subsidies, and education? Because these are the usual aspects that are always mentioned, but they all normally don't work. Education doesn't work because people don't live up in way. Taxation doesn't work because politicians just don't implement them, and subsidies doesn't work because how in the food system there are various actors circumventing the way where the subsidies should go. So all of the no options today.
You can be something within friends to solve such problems. But yeah, it's not so easy really to come up with a specific example. Think of cigarette smoking. The price of purchasing cigarettes was rich. People still buy cigarettes. Granted, it's
Like in Singapore, they don't even chew gum.
okay, really. So the price is very high.
No, really.
Okay, so that's the other thing as well. If you don't go the market route, then you see the non-market route where you actually just
In order to be fair to online people, we also have online questions. Maybe we can also try to answer that.
Yeah.
Because that discussion leads to very general topics. So it will be hard to answer.
So we have a question for you, Line. So Laura asked, she would be interested in hearing more about the 17 initiatives that you interviewed. Who were they? And did they cover different levels of the food system? And since you refer to consumption patterns and also production systems.
Yes, the initiatives were generally selected. There was a 4-step selection process, which I didn't really have the time to focus on. And generally, the idea behind was that they should all focus on all the 4 perspectives, ideally, of the project, meaning waste reduction, diet transformation, organic food and farming, as well as the overall system transition. So some of them were food banks, for instance, like in Warsaw in Poland. Some of them were, for instance, the associations like the Bayer District Association, Cilento, in Italy. So are being engaged in bringing about the change in ideally all these 4 perspectives of the project.
Okay, one last, and we really have to go on because
so I'll make it quick. So my question is more when you measure waste, how were you able to quantify? What kind of questions do you ask? And also, how did they respond to the questions? And also, in terms of measuring waste, was the survey geared towards measuring waste and also you take into consideration food waste and of the value chain or the processing of the value chain?
No, only, thank you for your question, only at the household level. And what I didn't have the time to mention was the exact methodology, how we did it. First of all, we relied on the previously conducted questionnaire on food waste, which was validated in terms of measurements, for instance, the spoon of something, and then gram equivalent, and perhaps a limitation of our specific survey in that regard was that it was a recall based, which means the respondents were actually asked about the previous week. So to kind of try to remember the entire week, what did you waste? And of course, it's subject to bias, and therefore it's not that exact. First problem. The second problem is, of course, the bias, because you would feel that it's social desirability is there. So you have to probably lower your actual waste. So that could have been also the problem with that approach. So, but it's otherwise difficult to ask people to measure the waste. So therefore, we had to rely on recall, and we did rely on recall for past week, also for the dietary questions as well. So it was a recall based, therefore I'm always emphasizing it's self-reported data. So of course, there is some bias there, and we can't say this is the exact measurement, the 399 per household and week.
yes, thank you very much. You asked for more funding. Just let me use this opportunity to
announce that we have a pre-announcement out from the Future Foods Partnership on Sustainable Food Systems, and that there will be a call probably open in November. So there will be some opportunities for food projects to get new funding, and this will only be the first one of 6 calls in the next 10 years. So there will be some opportunities to apply for new funding in the sustainable food area. So let's try if we can get the last talk, which should be an online talk. You are there? Okay. Let's try if I open this here, if we can hear you as well. Can you say something? Okay. Okay, then we have to use your microphone as well again. How do we do it with your presentation? Shall I share your presentation or? Yes, right. And I will do that. It's here.
Okay, that's me. Good.
Yes, I will just switch off my microphone so that we don't have this echo, and then you can start.
Okido. So now I will indicate when I wish to get to the next slide. So I want to talk in the next 10 minutes about Bio4Food. This project is finished. We submitted the final report this September. And next slide. So we had 4 main objectives, which we did not completely succeed to fulfill, but we are contributed to these objectives. And what we have done is to try to reduce crop waste. We have contributed to circularity of the nutrient cycle. We have looked into crop resilience under abiotic and biotic stresses using these biostimulants and biopesticides that I mentioned during my pitch talk. And we have found conditions in which we can enhance mineral nutritional quality of vegetable crops. So these 4 objectives, we try to achieve these by valorizing vegetable and herbal waste and convert them into biostimulants and biopesticides. Next slide. This is the project overview with 5 work packages. In the first work package, we have been collecting the waste material that we wanted to test and use. And this was done both from waste that was coming from industry as well as waste that was collected from harvest material. And industry, I mean the food industry, as well as industry that extracts plant material for medicational or other purposes. In work package 2, we collaborated with ILU and Omnichem, the company that is involved in this project, to make extractions. And in total, we have made 45 extractions in work package 2. In the work package 3, the aim was then to start using these materials to test in various settings. And in these, I will explain a little bit later what we did. So both in vitro, pot experiments, field experiments. And then we choose a number of products specifically to increase the production so that we could come closer to trying to develop commercially viable products. And then we tested them again and developed a roadmap and socioeconomic study to evaluate the potential implementation of our products. Next slide. These are the sources that we've used. This included 8 herbal and other crop wastes, and you can see mint, you can see rosemary, you can see basil, artichoke, parsley, rosemary, tomato, and fennel. And there were also 3 sources of waste material that we got from a company, Omnichem, that were high in tannin content. Next slide. As I said, these wastes were then extracted using different methods, mainly water and ethanol. This is because other products that other extraction methods are not allowed by the European legislation for production of biostimulants. So we were in line with this regulation. And so the 11 waste streams were converted into 45 extracts. And then they were redistributed to CREA and Ghent University to test them for bioactivity. And at CREA in Italy, Bari, they were also using these materials for composting. So this was done with the original material, but also by using the extracts. Next slide. In work package 3, we validated crop performance. We've done a large number of tests that are at 3 levels. They were in vitro tests, looking into root growth and shoot growth. We also did plate testing for fungicidal and nematicidal activity. At CREA, they did a number of pot experiments. That means you put your plants in a pot using a substrate, and these substrates were complemented with compost or with biostimulant or biopesticide. And then with some field trials, we did that at Ghent University and at CREA. And we tested, for example, a 2-year rotation of fennel tomato, looking into the yield and nutrient concentrations. And here the nutrient concentrations, I mean the minerals, iron, zinc, magnesium, and copper in the crop, the final crop that you eat. Also, a winter crop fennel was done using parsley extract and a spring summer tomato using basil extract. Next. Next slide. Yeah. So what did we find? Well, actually, we found quite a lot. I did not list the whole series of experiments that we've conducted, but I think here I highlight 3 topical results that stood out compared to the others. And let me start with what we found in my own team, in my own research lab. So we identified a product, it's called Omnivin R, which is a waste material that is generated by the company Omnichem. And we found that it has strong fungicidal activity next to other tannin extracts, in fact. We also found a nematicide activity, but we did not find biostimulant activity. And in the graph, you can see blue bars, 2 blue bars that are very high, one indicated with a black arrow. That is the biological activity against nematodes identified in our tannin extract. And you can compare it with the efficiency that is obtained with the reference molecules, Vertimec, or the reference product, used by the farmers to kill nematodes. So you see that we nearly reached the same level of efficacy. And in the photograph, you can see nematodes in white as white lines, and when they are completely straight, they basically are dead. At CREA in Italy, they have tested a number of extracts and composted materials, and here they found actually that by using parsley extract, they could stimulate the concentration of nutrients present in the vegetables. So demonstrating that using extracts from waste materials can actually promote or improve crop quality and the food quality. At Morocco in FTTS, they were testing mainly rosemary extracts because they have a lot of this rosemary extract waste that comes from harvesting the crop, and the twigs that are not being used, there's massive amounts that they can extract using water and ethanol. And they have been applying this to test against a whole range of fungi. And here they demonstrated that they could inhibit botrytis infection and infestation on harvested tomato by treating these tomatoes with a rosemary extract. Next slide. Here, if you could go and click with your mouse at the bottom of the slide, you will, you can move, and you will normally activate a movie. Maybe you have to click into it. I see it's not popping up. Can you click in the middle of the black screen? Does it do anything? No, doesn't recognize it. Okay, I will continue explaining what I hope that you could see. It's basically a drone flying over a field of Brassica napus, grown here in the vicinity of our university. And we had applied different products, different extracts that we obtained from the waste streams. And these extracts showed minor growth promotion using in vitro assays. And so we wanted to test whether these growth promoting effects were also identified by applying them into the field. And basically what we found is that these extracts did not affect, they did not change the total yield of seeds coming from brassica napus or rapeseed. So this was, of course, a negative result, if you like, but then still we were quite happy that these products did not show a growth inhibition. Further experimentation will be required to test whether there should be different concentrations or different moments of application to see any growth impact. Next slide. So we did also quite a lot of outreach. We organized first international conference, Sustainable Agriculture and Biopesticides and Biostimulants. That was in 2022 here in Ghent, where we had 125 participants, of which half of it came from the industry. And there we presented the research report on the Bio4Food project. We've also collaborated with ILVO, a local research station, on a market study and published an article, it's in Dutch, but still, on biopesticides and biostimulants about opportunities and challenges for the Flemish bioeconomy. We've also posted a document on the practical use of compost. This was done by CREA. We also participated to a Biostimulant World Congress, in fact 3 times during the course of this project. And now, next in November, we will again present our work on Bio4Food at this Congress. We participate to Ferti and Growth. This is an electronic platform for adult learning in Europe. And we are contributing to an online tool to study or to get information on biostimulants. It's called biostimulant.com or the Biostimulant Academy. Next slide. So what can we now do with the results we have obtained? So we are prospecting the agro industry for developing this nematocidal product. And since it is originally coming from Ajinomoto, we are collaborating with them. They have created a formulated unenriched biopesticides for a future field trial. We're evaluating the patenting of the nematicide from the waste extract. And this will depend on further purifications of the material into pure chemicals and whether they can be shown to be effective against the nematodes. We're prospecting the agro industry for developing a fungicide product. We have posted and continue to do recommendations through CREA for the use of specific waste material for composting and how this can be used in selectable vegetable crop rotation systems. And then at FSTT in Morocco, they are consulting legal entities to assess the registration and use of rosemary extract as a fruit preservative. Next slide. This is the end of my presentation, and I would like to acknowledge the Bio4Food team. It has been a very smooth collaboration between the different partners despite the COVID period. I also want to thank the CropFit team. CropFit is a local business unit from Ghent University that supports the valorization of the results that we have obtained. Technical support, of course, from local people, and then my collaborators, Jing Li and Thijs van Gerwey, who have mainly performed these research activities and coordinating everything else. With that, I thank you, and I'm ready for questions.
Yes. Are there any questions in the audience? Yes.
Should I speak up? Yeah.
So the waste streams that you mentioned is not your general waste streams. They seem to be quite specific. How do you secure them, especially if you are looking at developing a product and commercializing? How do you secure those specific waste streams?
Okay, so I think I heard the question, how did we select the specific waste streams? So a number of criteria have to be fulfilled to be suitable for a biostimulant and a biopesticide. The first is that the waste stream should be in sufficient quantities. Second, it should be very inexpensive, and it should be immediately available. I give you an example. There's a lot of waste left in the fields. If you have to go and collect this non-harvested material from the fields to convert it into a biostimulant, that will be too expensive. So therefore, you've seen many herbs that were selected. And this is because we were in contact with a company that freeze dries these herbs, but they cut away a lot of the leaf material. And this is waste they have to get rid of. And it's already frozen, so it's actually conserved. So this was ideal material to use for extraction. Secondly, we want to use mostly materials that are like very far in the development of the plant. When you have a plant that is exposed to the environment and to diseases and abiotic stresses, they typically accumulate a lot of defensive molecules, and especially at the outside of the tissue. So for example, in the epidermal cell layer. So what you want is high levels of epidermal cells in your material. So if you take a potato, for example, it will be very useful to use the potato peels, but it will be very not useful to use the central part of the potato because there are no defensive molecules to be found there. So this is how we decided to make a list of products. And then we also used industrial waste from this extraction company that uses plant material for making a whole range of products that are sold for food, but they have a lot of waste which they need to get rid of. They have to pay for removing this waste. So therefore they were quite keen on seeing any valorization for this waste. Yeah, that's my response.
Okay, I could see there was one online question to you. The extracts, were they formulated? How did you formulate them?
Yeah. So this is a very important question. Formulation is typically well developed for hydrophobic molecules. Most pesticides and defense molecules that are used currently by the industry are hydrophobic molecules, and they are emulsified in water using emulsifiers. But here we have used water and ethanol extracts. So most of these very hydrophobic molecules are not in our products. So we could just use directly the water solubilized or ethanol solubilized materials and spray them on the plants or water the plants with it. The risk that this would not be active is unjustified because former experiments project that I conducted showed that more than half of the biologically active molecules are highly water soluble. This is an underexplored group of molecules simply because the current the industry that is focused on agrochemicals has never properly exploited them.
Okay, thank you very much. I'm afraid I have to cut off everything here because everybody's standing outside drinking coffee already and people are getting nervous. So we also need to have a little bit of coffee break. So thank you very much to all presenters and everybody.
Bye.
We will meet here again at 16:10, so 10 minutes past 4.